Hello dear students, today our topic of deliberation is general properties of viruses, cultivation of viruses, virus purification and assays. Before going through this topic, first let's have a look on its objectives. Number 1, general properties of viruses. Number 2, cultivation of viruses. Number 3, virus purification and assays. Now let's start with the first objective, which is general properties of viruses. Viruses are a unique group of infectious agents whose distinctiveness resides in their simple acellular organization and pattern of reproduction. A complete virus particle or virion consists of one or more molecules of DNA or RNA enclosed in a coat of protein and sometimes also in other layers. These additional layers may be very complex and contain carbohydrates, lipids, and additional proteins. Viruses can exist in two phases, extracellular and intracellular. Virions, the extracellular phase, possesses few enzymes and cannot reproduce independent of living cells. In the intracellular phase, viruses exist primarily as replicating nucleic acids that induce host metabolism to synthesize virion components. Eventually, complete virus particles or virions are released. To summarize, viruses differ from living cells in at least 3 ways. Number 1, their simple acellular organization. Number 2, the presence of either DNA or RNA, but not both, in almost all virions. Human cytomegaloviruses have a DNA genome and 4 messenger RNA molecules. Number 3, their inability to reproduce independent of cells and carry out cell division as prokaryotes and eukaryotes do. Now, let's discuss the second objective, which is cultivation of viruses. Because they are unable to reproduce independent of living cells, viruses cannot be cultured in the same way as bacteria and eukaryotic microorganisms. For many years, researchers have cultivated animal viruses by inoculating suitable host animals or embryonated eggs, fertilized chicken eggs incubated about 6 to 8 days after laying. To prepare the egg for virus cultivation, the shell surface is first disinfected with iodine and penetrated with a small sterile drill. After inoculation, the drill hole is sealed with gelatin and the egg is incubated. Viruses may be able to reproduce only in certain parts of the embryo. Consequently, they must be injected into the proper region. For example, the myxoma viruses grow well on the chorioallantoic membrane, whereas the mumps virus prefer the allantoic cavity. The infection may produce a local tissue lesion known as pock, whose appearance often is characteristic of the virus. More recently, animal viruses have been grown in tissue culture on monolayers of animal cells. This technique is made possible by the development of growth media for animal cells and by the advent of antibiotics that can prevent bacterial and fungal contamination. A layer of animal cells in a specially prepared petri dish is covered with a virus inoculum, and the viruses are allowed time to settle and attack to the cells. The cells are then covered with a thin layer of agar to limit virion spread so that only adjacent cells are infected by newly produced virions. As a result, localized areas of cellular destruction and lysis called plaques often are formed and may be deducted if stained with dyes such as neutral red or type in blue that can distinguish living from dead cells. Bacterial viruses or bacteriophages are cultivated in either broth or agar cultures of young, actively growing bacterial cells. Agar cultures are prepared by mixing the bacteriophage sample with cool liquid agar and a suitable bacterial culture. The mixture is quickly poured into a petri dish containing a bottom layer of sterile agar. After hardening, bacteria in the top layer of agar grow and reproduce, forming a continuous opaque layer or LAN. Wherever a virion comes to rest in the top agar, the virus infects an adjacent cell and reproduces. Eventually, bacterial lysis generates a plaque or clearing in the lawn. Plaque appearance often is characteristic of the phage being cultivated. Now students, you know, plant viruses are cultivated in a variety of ways. Plant tissue cultures, cultures of separated cells, or cultures of protoplasts may be used. Viruses also can be grown in whole plants. Leaves are mechanically inoculated when rubbed with a mixture of viruses and an abrasive such as carborundum. When the cell walls are broken by the abrasive, the viruses directly contact the plasma membrane and infect the exposed host cells. A localized necrotic lesion often develops due to the rapid death of cells in the infected area. Even when lesions do not occur, the infected plant may show symptoms such as change in pigmentation or leaf shape. Some plant viruses can be transmitted only if a diseased part is grafted onto a healthy plant. Now coming to the third objective of this topic, which is virus purification and assays. Virus purification makes use of several virus properties. Virions are very large relative to proteins, or often more stable than normal cell components, and have surface proteins. Because of these characteristics, many techniques useful for the isolation of proteins and organelles can be employed in virus isolation. Four of the most widely used approaches are: number 1, differential and density gradient centrifugation; number 2, precipitation of viruses; number 3, denaturation of contaminants; number 4, enzymatic digestion of cell constituents. Now let us discuss the number 1. Host cells in the later stages of infection that contain mature virions are used as the source of material. Infected cells are first disrupted in a buffer to produce an aqueous suspension, or what we call homogenate, consisting of cell components and viruses. Viruses can then be isolated by differential centrifugation, the centrifugation of a suspension at various speeds to separate particles of different sizes. Usually, the homogenate is first centrifuged at high speed to sediment viruses and large cellular particles, and the supernatant, which contains the homogenate's soluble molecules, is discarded. The pellet is next resuspended and centrifuged at a low speed to remove substances heavier than viruses. High-speed centrifugation then sediments the viruses. The process may be repeated to purify the virus particles further. Viruses also can be purified based on their size and density by the use of gradient centrifugation. A saccharose solution is poured into a centrifuge tube so that its concentration smoothly and linearly increases between the top and the bottom of the tube. The virus preparation, often after purification by differential centrifugation is layered on the top of the gradient and centrifuged. The particles settle under centrifugal force until they come to rest at the level where the gradient's density equals the Earth's. Now let us discuss the second one. Viruses, like many proteins, can be purified through precipitation with concentrated ammonium sulfate. Initially, sufficient ammonium sulfate is added to raise its concentration to a level just below that which will precipitate the virus. After any precipitated contaminants are removed, more ammonium sulfate is added and the precipitated viruses are collected by centrifugation. Viruses sensitive to ammonium sulfate often are purified by precipitation with polyethylene glycol. Number third, viruses frequently are less easily denatured than many normal cell constituents. Contaminants may be denatured and precipitated with heat or a change in pH to purify viruses, because some viruses also tolerate treatment with organic solvents like butanol and chloroform. Solvent treatment can be used to both denature protein contaminants and extract any lipids in the preparation. The solvent is thoroughly mixed with virus preparation, then allowed to stand and separate into organic and aqueous layers. The unaltered virus remains suspended in the aqueous phase while lipids dissolve in the organic phase. Substances denatured by organic solvents collect at the interface between the aqueous and organic phases. Number 4, cellular proteins and nucleic acids can be removed from many virus preparations through enzymatic degradation, because viruses usually are more resistant to attack by nucleases and proteases that are free nucleic acids and proteins. For example, ribonuclease and trypsin often degrade cellular ribonucleic acids and proteins while leaving virions unaltered. Now, dear students, let's discuss virus assays. The quantity of viruses in a sample can be determined either by counting particle numbers or by measurement of the infectious unit concentration. Although most normal virions are probably potentially infective, many will not infect host cells because they do not contact the proper surface site. Thus, the total particle count may be from 2 to 1 million times the infectious unit number, depending on the nature of the virion and the experimental conditions. Despite this, both approaches are of value. Virus particles can be counted directly with the electron microscope. In one procedure, the virus sample is mixed with a known concentration of small latex beads and sprayed on a coated specimen grid. The beads and virions are counted. The virus concentration is calculated from these counts and from the bead concentration. This technique often works well with concentrated preparations of viruses of known morphology. Viruses can be concentrated by centrifugation before counting if the preparation is too dilute. However, if the beads and viruses are not evenly distributed, the final count will be inaccurate. The most popular indirect method of counting virus particles is the hemagglutination assay. Many viruses can bind to the surface of red blood cells. If the ratio of viruses to cells is large enough, virus particles will join the red blood cells together, forming a network that settles out of suspension or agglutinates. In practice, red blood cells are mixed with a series of virus preparations, and each mixture is examined. This assay is an accurate, rapid method for determining the relative quantity of viruses, such as the influenza virus. A variety of assays analyze virus numbers in terms of infectivity, and many of these are based on the same techniques used for virus cultivation. For example, in the PlaQ assay, several dilutions of bacterial or animal viruses are plated out with appropriate host cells. When the number of viruses plated out are much fewer than the number of host cells available for infection, and when the viruses are distributed evenly, each PlaQ in a layer of bacterial or animal cells is assumed to have arisen from the reproduction of a single virus particle. Therefore, a count of the PlaQs produced at a particular dilution will give the number of infectious virions or PlaQ forming units, and the concentration of infectious units in the original sample can be easily calculated. Suppose that 0.10 ml of a 10⁶ dilution of the virus preparation yields 75 PlaQs. The original concentration of PlaQ forming units viruses producing different PlaQ morphology types on the same plate may be counted separately. Although the number of PFU does not equal the number of virus particles, their ratios are proportional. A preparation with twice as many viruses will have twice the PlaQ forming units. The same approach employed in the PlaQ assay may be used with embryos and plants. Chicken embryos can be inoculated with a diluted preparation, or plant leaves rubbed with a mixture of diluted virus and abrasive. The number of pokes on embryonic membranes or necrotic lesions on leaves is multiplied by the dilution factor and divided by the inoculum volume to obtain the concentration of infectious units. When biological effects are not easily quantified in these ways, the amount of virus required to cause disease or death can be determined by the end-point method. Organisms or cell cultures are inoculated with serial dilutions of a virus suspension. The results are used to find the end-point dilution at which 50% of the host cells or organisms are destroyed. The lethal dose is the dilution that contains a dose large enough to destroy 50% of the host cells or organisms. In a similar sense, the infectious dose is the dose which, when given to a number of test systems or hosts, causes an infection of 50% of the systems or hosts under the conditions employed. This brings us to the end of lecture. Hope you have enjoyed and understood. Thank you and goodbye.